Hi everyone, it's Fiona again from the NCBE. Thank you for joining me. So today we're going to be learning all about DNA. So for those of you that have already studied this in your A-level syllabus, hopefully this will be a useful revision aid. And for those of you who haven't, hopefully we'll learn something new. Okay, so what are we actually going to go over today? So we're going to have a look at the structure of both DNA and RNA and the differences and similarities between them. We're also going to have a little look at the molecule ATP, since this is very important in our bodies, and it's actually very similar to an RNA nucleotide. We'll then move on to looking at the exciting history behind the discovery of DNA and some of the key experiments and scientists that worked on this in the lead up to Watson and Crick unveiling the three-dimensional structure of DNA. So DNA, or deoxyribonucleic acid, is needed for the long-term storage of genetic information in every cell in our bodies. This is arranged into chromosomes that are stored in the nucleus. So ribonucleic acid, or RNA, on the other hand, comes in several forms. It comes as messenger RNA, where it acts as a messenger molecule, taking a copy of DNA over to the ribosomes to be translated into proteins. It also is found actually in the ribosomes as ribosomal RNA, and very importantly, it is found as transfer RNA, where it brings the amino acids over to the ribosomes when messenger RNA is being translated. So both DNA and RNA are very similar structurally. They're both made up of a polymer of nucleotides, and these nucleotides include a pentose sugar, a phosphate group, and a nitrogenous base. Now, the pentose sugar and the phosphate group make up the backbone of either the DNA or RNA molecule, and the nitrogenous bases are connected to the pentose sugar. So in DNA, these nitrogenous bases are adenine, cytosine, guanine, and thymine. And in RNA, they are adenine, guanine, cytosine, and uracil. So the nucleotides are connected by phosphodiester bonds, and these form by condensation reactions. So you can see here that both RNA and DNA use two purine bases, and these are adenine and guanine in both. And they also use two pyrimidine bases that are cytosine in both, or are thymine in DNA or uracil in RNA. The main differences between DNA and RNA then are that DNA has deoxyribose as its sugar molecule and RNA has ribose. Now they're actually still pretty similar, the two sugars. The only difference really being that deoxyribose has one less oxygen molecule in it compared to ribose. And you can see that here. So they also differ then in the nitrogenous bases that they use. So DNA, as I mentioned, uses thymine and RNA uses uracil. And thymine and uracil are also pretty similar in structure. The only real difference is that uracil is lacking one methyl group that is available in thymine. Lastly then, and this really is the most obvious difference between DNA and RNA, is DNA is obviously double-stranded and therefore forms a helix, and RNA is single-stranded. DNA is also much, much longer than RNA, since it obviously stores all of the genetic information that we need in our bodies, whilst RNA tends to be much, much shorter. So in DNA, the two antiparallel strands align, and the nitrogenous bases form hydrogen bonds between the complementary base pairs. So G always binds with C, and it does this with 3 hydrogen bonds. And A always binds to T, and it does this with 2 hydrogen bonds. This causes a double-stranded structure, which then twists into a helical arrangement. So this complementary base pairing means that the proportions of G and C are always the same, and the proportions of A and T are always the same, meaning that actually if you get asked this in an exam to calculate the composition of a particular DNA molecule, and you've been told the proportion of one base, you can actually work out the proportions of all the other bases. So for an example, if you know that you have 25% adenine, then you must also have 25% thymine, because obviously they are complementary base pairing with each other. That then means you have 50% accounted for and 50% still account for. Well, then all you need to do is divide 50% by 2, which obviously leaves you 25% guanine and 25% cytosine. So that would mean in this example that all the bases have equal proportions. Okay, so before we move on to then the history of DNA, I'll just quickly show you the structure of ATP, since as I mentioned, this is obviously a very important molecule in our bodies. And you can see here on this diagram how similar it is to an RNA nucleotide. So it has a ribose pentose sugar, same as in RNA. It has a nitrogen-containing base and an ATP that's always adenine. And it also has a phosphate group attached. Now, instead of there being just one phosphate group, as there is in an RNA nucleotide, there's 3 in ATP. And these phosphate groups are what allows ATP to act as an energy source in the cell. So when the phosphate anhydride bond is broken and a phosphate molecule is released, then that causes energy to be released, which is how different chemical reactions are powered within the cell. So ATP is then converted to ADP, which has 2 phosphate groups. And ATP can then be recycled, and this phosphate group can be added back on again, and it can be used again. Okay, so let's now move on to looking at the history of DNA and how it was discovered. So here's a small timeline of some key events that led to the discovery of DNA and the understanding that it was DNA that was the important genetic material that allowed inherited traits to be passed on from one generation to the next. So let's start with Gregor Mendel then in 1865. So he presented his data about how inherited traits are passed from one generation to the next, and he did this using pea plants. So at this time, previously, inheritance was thought to occur by a process of blending. Now this was really because mostly traits such as height, which are obviously continuous traits, have been looked at. And you can sort of see how they might have come to that conclusion. So if you, for example, had a tall parent and a smaller parent, then perhaps the offspring would have a height of somewhere in between. So they thought that these traits were just blended together and then passed on to their offspring. So Gregor Mendel looked at discontinuous traits that had discrete outcomes. So using his pea plants then, he looked at, for an example, flower colour, the texture of their seeds, and these had, as I said, discrete outcomes. So for an example, the seeds were smooth or they were rough. And by doing this, he was able to deduce which traits were dominant and which traits were recessive by looking at multiple generations of these pea plants. So importantly, Mendel showed that traits were inherited from one generation to the next in specific patterns, and this disproved the theory of blended inheritance. Now, although Mendel had obviously discovered how traits were passed on from one generation to the next, the molecule which actually caused this, DNA, hadn't actually been discovered yet. So in 1869, Friedrich Miescher identified what he termed nuclein by isolating a molecule from a cell nucleus. And this is what was later then became known as DNA. So he is really known then for first discovering the molecule we call DNA. However, it wasn't actually for many years that people really started to understand that it was DNA that carried the genetic information and how it did this. So many scientists then over the next 80 to 90 years worked on understanding inheritance and what the molecule was that carried the genetic code. So for a while people thought that proteins were the genetic material because they seem much more complex than DNA. DNA just seemed too simple with only 4 bases and apparent repeating sequences. There were a few discoveries that brought us closer to understanding DNA, and I'll just mention a couple of key ones before we move on to Watson and Crick's cracking the 3-dimensional structure of DNA. So firstly, Chargaff investigated DNA from different species, and he discovered that the proportion of the bases was not equal within a species. So there wasn't 25% A, T, C, and G. They varied. However, very importantly, what he did discover was that the proportion of A was always the same as T, and the proportion of G was always the proportion of C. And this, as we will see in a minute, was obviously very critical to Watson and Crick cracking the 3-dimensional structure of DNA. Now, knowing that these bases always match each other in proportions became known as Chargaff's rules. So next then, we move on to Rosalind Franklin, who, along with Maurice Wilkins, analysed DNA using X-ray crystallography. Now, they produced some incredible images of all different sorts of molecules using this technique. But very importantly, they produced some very clear images of DNA that when you look at the X-ray diffraction images, and when they were analysed later by Watson, you can start to understand that actually DNA is a double helix. So then let's move on to Watson and Crick. So in 1953, Watson and Crick put together these findings from both Chargaff and Rosalind Franklin. And they came up with their three-dimensional structure of DNA. And obviously, along with Wilkins, we know this led to them winning the Nobel Prize for Medicine in 1962. So by understanding that the proportions of A and T and G and C always match, they understood this showed the complementary base pairing system we know is found in DNA. And the diffraction pictures taken by Franklin showed that the structure was likely two-stranded and helical. So when they started to make models using these criteria, they cracked the mystery that so many have been working on. This discovery also really opened the door to many further discoveries in the fields of genetics and molecular biology. Understanding the structure of DNA also led to the understanding of how DNA replicates, which was hinted at by Watson and Crick, but wasn't fully understood until 1958, when scientists Messelson and Stahl published their seminal experiment. So they showed that DNA replicates semi-conservatively. So this means that at the end of each DNA replication cycle, you will have an original strand paired with a new strand, and another original strand paired with a new strand, meaning that the original strands have acted as a template for the new strands to form. And this is instead of conservative replication, in which two original strands would be paired with each other and two new strands would be paired with each other after each DNA replication cycle. So this was another theory that was around at the time. So how then did they actually show that DNA replicates by semi-conservative replication? Well, as you can see from my diagram here, they did this using bacteria. So they supplied a bacterial culture with nitrogen-14, which is the most common nitrogen that was normally found in the environment. And they also gave another flask of bacteria nitrogen-15 as their main nitrogen source. Now, nitrogen is needed in DNA replication because obviously all the nitrogenous bases in DNA need nitrogen. So if the only nitrogen available is nitrogen-15, that is what will be taken up into the DNA when it is being replicated, and the same for nitrogen-14. So those that would have nitrogen-15 as their main nitrogen source would be heavier than those with nitrogen-14. So you can see in my diagram I've got an orange culture representing the nitrogen-14 media and the red culture showing the nitrogen-15 media. So what they then did was extract the DNA from both cultures and spun them very, very fast in an ultracentrifuge in a solution containing cesium chloride, which, when it's spun at very high speeds, allows a density gradient to form. So this resulted in a higher band for those that had been grown in nitrogen-14 because it's lighter, and a lower band on the test tube for those that had been grown in nitrogen-15 because obviously it's heavier. So what they then did was they took some of the bacteria that had been grown in the nitrogen-15 culture and they put it into media containing nitrogen-14. Now they allowed them to go through one round of DNA replication, and they then took some of the bacterial culture out, again extracted the DNA, and again then spun it very, very quickly in a centrifuge in this density gradient. And what they very interestingly found was a band in between the nitrogen-14 band and the nitrogen-15 band, showing that the weight of that DNA was in between that of nitrogen-14 and that of the bacteria that had been grown in nitrogen-15. Now, this proved that the DNA was replicating by semi-conservative replication, because if it had been using conservative replication, you would have actually had two bands, wouldn't you? Because you would have had the DNA, the two original strands, which would have been still heavy in nitrogen-15, and the two new strands would have then been lighter, having used the new nitrogen-14 that was available. But instead they didn't see two bands, they saw the one in the middle, showing that this DNA was in between the weight of those from nitrogen-14 and nitrogen-15. Okay, so let's just recap then. So today we have looked at both the structure of DNA and RNA, and we've looked at the similarities and the differences between the two. We've also had a little look at the molecule ATP and how this is quite structurally similar to an RNA nucleotide. We then moved on to looking at the history of DNA and the key scientific experiments which then led up to Watson and Crick discovering the structure of DNA. So I'm going to leave it there for this video, and you can sign in for part 2, in which we will be covering DNA replication and also how errors in replication can lead to mutations. And we will have a look at cystic fibrosis as an example of a genetic disease. Thank you.